right, my friends, welcome back. This is going to be part 8 of my presentation on the microscopic world. And in this one here, guys, we're going to be talking about proteins and amino acids. Proteins and amino acids. If you'll notice here, let's start off with a definition. A definition of protein: any of a large number of organic compounds that make up living organisms and are essential to their functioning. First discovered in 1838, proteins are now recognized as the predominant ingredients of cells, making up more than 50% of the dry weight of animals. The word protein is coined from the Greek proteos, or primary. All right, let's take some notes on proteins here. Humans have an estimated 30,000 different proteins. About 2% have been described. Fibrous proteins. You want to think of fibers, tissues, collagen, keratin, fibrinogen, muscle proteins. Collagen makes up bone, skin, tendons, and cartilage, most abundant protein. Keratin makes up the outermost layer of skin and hair, scales, hooves, nails, and feathers of animals. Fibrinogen is a blood plasma protein responsible for blood clotting, and we're going to be talking about that here real soon. It's towards the end of this presentation. I think it's like the last block. It's next to the last topic of this presentation. We're going to be getting into blood clotting, which is absolutely just amazing. Muscle proteins is responsible for muscle contraction. Globular proteins, spherical or globe-like, enzymes, protein hormones, antibodies, microtubules. Enzymes, round proteins that combine with other substances called substrate to bring about numerous chemical reactions in the body, many responsible for metabolism or energy conversion. And that metabolism is breathing, circulating blood, controlling body temperature, contracting muscles, digesting of food and nutrients, eliminating waste, functioning of the brain and nerves. Now, protein hormones, this is called messenger proteins. They stimulate target organs to produce insulin, control of blood sugar, oxytocin, controls contractions during childbirth, somatotropin. I'm going to try that one more time. Somatotropin. It's a growth hormone that stimulates protein production in muscle cells. Antibodies, an important component of the immune system and act primarily as a defense against foreign substances. The manufacture of antibodies is initiated when a foreign substance, antigen, like bacteria and viruses, enters the body. Lymphocyte cells respond by making an antibody with a molecular arrangement that fits the shape of molecules on the surface of the invader, so that the antibody combines with it, and either one, weakens it so other blood proteins can attack, or two, directly inactivating, and three, by allowing other blood cells to come in and destroy them. And guys, all this supposedly came by blind chance. And so, microtubules, hollow tubes that serve both to structure cells and to conduct substances from one part of the cell to another. Microtubules also make up the inner structure of cilia. And we're going to talk about cilia also later. The hair-like appendages by which some microorganisms propel themselves. And so, yes, we are going to talk about that little area there. The propelling of themselves with is absolutely just astounding. Okay, guys, it's time for chalkboard. You got the chalkboard time with me again. And so we're going to look at amino acids. amino acids. Now, you got 26 letters in the alphabet. 26 letters in the alphabet. Now, supposedly, this sentence right here has all the letters in the alphabet. The quick brown fox jumps over the lazy dog. The quick brown fox jumps over the lazy dog. Suppose it has all the letters of the alphabet inside of it. Now, protein amino acids, the building blocks of proteins. And so amino acids, basically amino acids, guys, you're going to hear that over and over and over again. Some of you guys that are into exercise, building, bodybuilding, stuff like that, you know what I'm talking about, because a lot of these health food stores make a big deal over amino acids. Those you'll hear that on these protein shakes and stuff. And protein, those are words you're going to see a lot. And but at any rate, these are amino acids right before you. And see those letters there, guys? Think about that. Let's go back to that one slide here, 26 letters in the alphabet. So you're going to think of amino acids like letters in an alphabet. Letters in an alphabet. So let's go back here. You got all these amino acids here. I'm not going to go through all of them, but look at the letters. Look at the letters on the left-hand side of the amino acids. You're going to think of those letters as the alphabet, okay? Now, 11 are non-essential. 11 are non-essential, which means the body can produce them and they are not required in a diet. So your body can produce these amino acids right here. They produce them all, by itself. 9 are referred to as essential. The body cannot produce them. They have to be taken in as food from meat, fish, milk, and eggs. These proteins are called complete proteins. Now, what is interesting here, guys, I'm going to kind of just rabbit trail here just a minute. Before the flood, everybody ate vegetables. And it's possible, guys, that these as 9 essential ones, they were somehow produced in nature. And with the fall, they were not produced anymore. Or maybe we just didn't need these. We just didn't need these in our body. After the fall, though, God told them they could eat meat now. They can now eat meat. And so now we do have to have these in our diet now. Now, amino acids are like letters in a sentence. The quick brown fox jumps over the lazy dog. So you think of that L there, that is one amino acid. Just like the letters in a sentence, the amino acids must be in the right sequence to bond together. So you would think of the whole sentence as a protein. The whole sentence is a protein. The smallest known protein has 70 to 100 amino acids. Think about that. So one letter out of this sentence is an amino acid. The whole sentence is a protein. And you get a whole bunch of sentences that come together to make paragraphs, and you got the cell. So all these paragraphs here would be considered a cell. That would be considered a cell. Or to put it another way, you would think of like the Morse code. Think of like the Morse code. You know that dot, 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 you've heard of that before. And they code for certain letters. And those letters make certain words. And the words, you can actually make sentences, paragraphs, and you could write books out of those words. Just like that, our DNA will code for the amino acids, which is those amino acids are like letters in sentences. You would think of those sentences as proteins, and a bunch of proteins make up us and living organisms and stuff like that. You think about that. Guys, it's a whole lot more complex than you could ever imagine. There's no way we came about by blind chance. Now, let's talk about chromosomes. There are 23 pairs in the human cell. 23 pairs in the human cell. And you would think of the chromosomes are in the nucleus of the cell. Now, a normal human male, this is a male's chromosomes. Women have 2 X chromosomes, and men have 1 X and 1 Y chromosome. And so down there at the bottom right-hand corner, you can see that. Now, what is really amazing, do you guys know that, especially in like the Middle East, Um, it was men could, and I'm assuming they still can, they can divorce their wives if their wives could not produce them a son. But do you know that that is so wrong? Because the man is the one that is really responsible for the sex of the child. So in a turn, it's not the woman's fault, it's the man's fault. If the man doesn't, the man is the one that gives the sex to the child, not the woman. And so it just really is amazing when you come to think about that, how all those guys treated their women bad because they wouldn't produce a son, and it's not even the woman's fault at all. It has nothing to do with the woman. This type of cell is called a diploid cell. There are 23 pairs of chromosomes, or 46 chromosomes. There are 46 chromosomes. Reproductive cells like egg and sperm cells are called haploid or gamete cells. They only have 23 chromosomes, not pairs, just 23 chromosomes. Now, this is an ovum, or it's Latin, it means egg or egg cell. It's about the size of a grain of sugar. Now, there's the chromosomes right there. Now, it has a security wall, a gel-like shell around the egg. Now, the corona, I'm going to try this one, radiata. It's the blue areas that you are seeing. Look like little spots all around. That is what that area is. And this is a drawing of it, kind of give you an idea of what that is. Now, the cells unite with each other, combining each of the 23 chromosomes they carry to form a complete cell. So there's 23 chromosomes coming from the male and 23 chromosomes from the female. They come together to produce one complete cell, the male having the say-so on what the sex of the child is going to be. And so this is what we consider fertilization. It's now fertilization. This new cell is called a zygote cell, which is the first cell of an offspring. This union activates an unstoppable cell division process that produces an adult organism. Now, guys, now, if you want to see some science in the Bible, here's some science in the Bible. Genesis chapter 2, verse 24. Therefore shall a man leave his father and mother, and shall cleave unto his wife. And what does it say, guys? They shall be one flesh. Now, what happens in fertilization? You got 23 chromosomes from the man, 23 chromosomes from the woman. They come together, and what do they become? They become one person, one human being. And so there is more science in the Bible than you can ever imagine. Now, let's keep going here. Now, if you was to take a chromosome and start unwinding it, that's when you see, that's the DNA. That's the DNA. And so this picture right here, look at the bottom, there's your chromosome, and you just start unwinding it. Just start unwinding it, and eventually you're going to come to those strands of that DNA there. Now, let's code, genes code for protein structure. How does this happen? Excuse me. How does amino acids code for protein structure? Guys, I'm going to do the best I can to explain this. So get ready. You got right here where you got the gene and the DNA. You got, which is adenine, thymine, cytosine, guanine. So adenine goes to thymine and cytosine goes to guanine. And so these are what you call your base pairs, your bases. And so now DNA strands separate. So the DNA comes apart. Transcription begins. Construction of messenger RNA. Now that M means messenger. Messenger RNA. That's what that means. So the construction of messenger RNA, so it comes apart, and uracil is used by RNA in place of thymine. Now, guys, I'm not going to just try to define every little thing. I'm just giving you a basic overview here, okay? And I'm doing good to do that. Now, if you'll also notice that I've got circled up there codon. Codon, we're going to talk about that. Now, so this is what you got. Every 3 bases, now remember, we talked about the bases of the DNA. Every 3 bases code for 1 amino acid. They're called codons. Codons. So looking at it in a chart, this is what it would look like. So every 3 bases, they code for 1 amino acid. 1 amino acid. Now the removal of introns. And so using this chart right here, the introns are removed and messenger RNA is now ready. Messenger RNA binds to ribosome. So it comes out of the nucleus through those pores there. If you notice that picture up to the top right-hand corner, I got a red circle around it. That's what it comes out of. It comes out of there. And transfer RNA, that T, see that T there? That's transfer. Transfer, and I got that in parentheses, that tRNA, that's transfer RNA binds to amino acid. So, they go around and they start grabbing the amino acids. They start grabbing the amino acids that the messenger RNA says that it needs to build a protein, but it's going to be called a polypeptide chain. The translation sequence of amino acids is started. And so it starts going, it's going around in the cell and it's grabbing these amino acids and it's building this thing. And guys, this happens extremely fast, extremely fast. Ribosome is told to stop by stop codon. The manufacture of protein is complete. So you got that polypeptide chain there. There's your amino acids, but it ain't over with yet. It ain't done with yet. There's the polypeptide chain. Typical protein can have 50 to 3,000. Yes, I did say that. 3,000 amino acids. 50 to 3,000 amino acids. Now, the protein begins to fold. Now, this is where the miracle is. Now, I'm going to share with you some information here. At the end of the folding process, which typically takes anywhere from fractions of a second to a minute, 2 different proteins can be folded to structures as precise and different from each other as a 3/8 inch wrench and a jigsaw. And like the household tools, if their shapes are significantly warped, then they fail to do their jobs. Proteins like written languages or computer codes have a high degree of specificity. The function of the whole depends on the precise arrangement of the individual parts. Now, this guy right here is Christian B. Anfinsen, received his PhD degree in biochemistry from Harvard Medical School in 1943. He joined the National Institutes of Health in 1950. He's a Nobel Prize winner, guys. His research focused on understanding the relationship between a protein's 3-dimensional structure and its ability to function in a living cell. And this is what he says. The folding pattern can be as important as the amino acid units themselves, for without the necessary folding, the protein cannot react with other substances. In his studies of the enzyme ribonuclease, Anfinsen found that when he disrupted the linkages that hold in place the molecule's 3-dimensional structure, the enzyme became biologically inactive. This holds true even when the chain of amino acids remains intact. So that folding, and I'm going to give you a visual here of that folding. The folding of the amino acids into whatever protein it's supposed to be, if it doesn't, even though all the amino acids are together, it doesn't activate until it's folded. He demonstrated that proteins orient themselves into the most energetically stable formations. This brings certain amino acids close enough together to form the linkages that allow folding to take place. And Finson concluded that a specific and unique sequence of events must occur to produce proper folding in the protein. And again, guys, I want you guys to start thinking, this is going to happen by pure blind chance. Now, here, Francis Crick, we talked about Francis Crick in the last video. The most remarkable example of molecular architecture found in living organisms is undoubtedly the protein family. This intricate three-dimensional shape is essential for its function. Remember that, guys. To produce this miracle, now notice what he says, to produce this miracle of molecular construction, all the cell need to do is string together the amino acids which make up the polypeptide chain, which you know what that is now, in the correct order. This is a complicated biochemical process. Notice that. A molecular assembly line using instructions in the form of nucleic acid tape, the so-called messenger RNA. You know what that is. Here we need only to ask, how many possible proteins are there? If a particular amino acid sequence was selected by chance, how rare an event would that be? So, let's check it out. Francis gives this example. Take a protein that has 200 amino acids. This is a little less than an average small protein. By using 20 amino acids, remember, that's the letters in the sentence, you can get 20 to 200th power. I'm guessing that's how that's supposed to be said. 20 to the 200th power, different combinations. Guys, I'm about to show you how vast that is, which is approximately equal to 10 to the 260th power. 10 to the 260 power. Written out, this is what that would be. That's how many different combinations you can get by using 20 amino acids in a protein, 200 by using the 20 amino acids in a protein, 200 amino acids long. This is how many combinations that you can get. He goes on, these calculations take account only of the amino acid sequence. They do not allow for the fact that many sequences would probably not fold up satisfactorily into a stable, compact shape. So the folding, now the folding has to happen, which is also just as crucial. How rare? Typical evolutionists claim the universe is 20 billion years old. So 20 billion times 365 days times 24 hours times 60 minutes times 60 seconds, this is what you get. 630 quintillion, 720 trillion seconds. So you take that into that other number, and this is what you get. So what you get. That number right there, guys, this is how many changes per second for 20 billion years to make just one protein by unguided processes. This is for one protein, guys, with unguided processes that you got to get it right. Life did not come about by pure blind chance. What we have discovered is that even at this very basic level, there are complex structures which occur in many identical copies, that is, which have organized complexity and which cannot have arisen by pure chance. Life, from this point of view, is an infinitely rare event, and yet we see it teeming all around us. How can such rare things be so common? Guys, that came from Francis Crick, and I don't even know if he's a believer or not. I have no clue. So basically, you need a protein to get the cell, but you need the cell to get the protein. Basically, what we're saying is, it can't come about by pure blind chance. It had to be created. The cell had to have been created, spoken into existence by an almighty, powerful God, and then it starts to do its thing. There's no way. There's no way. You can't get the protein without the cell, and you can't have the cell without the protein. It can't happen. You cannot get, life cannot come about by pure chance, is what he's saying. So what we're saying is, this will give you a little idea about amino acids. This will be the last thing, and then we'll end this video from here. So just think, this right here, this is a toy that I think I got from a dollar store or something. Think of each one of these as amino acids, each one of them. And this is just a small one. Remember, they said the average is about 200 amino acids long. So imagine 200 of these things. Now, they're all together now. They've been sequenced, they've been transcribed, and you got them all together. Now, now you got what's called a polypeptide chain. It's not done yet. It won't function like this. It won't function like this. It's useless like this. Now it's got to fold. Now, the thing is, you don't just ball this thing up and say, okay, here you go. It's done. No. What's amazing is that the folding, it's like these amino acids have some type of reaction to each other, the way they're folded. I guess this touching this one like this, and then maybe this one coming over here and like that, and then this one coming down and touching that one like that, and then this one coming up like that, touching it on its end. Now it's in the shape that it has to be to function. Now it's turned on. This thing is now activated. Now what they did is they did a little experiment, and they interrupted this thing. They stopped, let's just say they stopped this red one from touching that blue one down here. It's inactivated. It's useless. It's useless. It couldn't do anything. And so that is how amazing this thing is, guys. And I cannot see how somebody that studies this stuff cannot be a Christian. I cannot, there's no way I could even understand that. I just can't. But like Jesus said, they have Moses and the prophets. If they don't believe this, guys, you can produce a miracle, which that's a miracle. And they won't even be persuaded. Why that is, it's just beyond me. It's beyond me. I would hate to be these people at the judgment, great white throne judgment. But guys, we're going to stop here. When we come back, we're going to continue on with this topic. And but I love you. You guys take care. Bye-bye.